Welcome, students. My name is Karen Lenisha Cabral, Assistant Professor in Botany at St. Joseph Vaz College, Kottalim. Today's lecture is for third year Botany students, Semester 5, paper code BOC 107, paper title Microbiology and Plant Pathology. Title of the unit Applications of Microorganisms. Module name Antibiotics, that is penicillin. Outlines Introduction, Types of antibiotics, Discovery of penicillin, Process of penicillin fermentation. Learning outcomes list the types of antibiotics, recalls discovery of penicillin, describes the process of penicillin fermentation. Introduction. What are antibiotics? Now, antibiotics is something that we commonly hear about in our day-to-day life with regard to in medical field. Now, antibiotics are substances produced by microorganisms that inhibit the growth and replication of bacteria. They do not work against viral infection, but they work only against bacterial infection. Now, when we talk of antibiotics, it depends on their mode of action. There are some antibiotics which are bactericidal, while there are other antibiotics which are bacteriostatic. Now, bactericidal antibiotics are those which kills the bacteria, while bacteriostatic are those which prevent the growth and reproduction of bacteria. Now, these are few types of antibiotics listed. We have penicillin, we have tetracycline, then we have quinolones, and many more. Coming to discovery of penicillin, how exactly was penicillin discovered? Now, the discovery of penicillin is attributed to Alexander Fleming. Now, there is a backstory to it. When Fleming returned to his laboratory from vacation, that is on September 28, 1928, he noticed some petri dishes containing Staphylococcus bacteria contaminated with a blue-green mold called Penicillium notatum, which was preventing the normal growth of bacteria. He obtained an extract from this mold, named its active ingredient as penicillin, and determined that the extract killed many types of harmful bacteria that infected humans. By discovering this mold, Fleming created one of the most powerful drugs in medical history. Now, penicillin are widely used in treatment for a variety of bacterial infections of skin, then chest, as well as urinary tract. Now, when penicillin is given to patients with an infection, it kills or prevents the bacteria from growing and multiplying by inhibiting the bacterial cell wall synthesis, thereby allowing a person to recover from the infection. Now, here there are some types of penicillin which are noted. We have penicillin G, penicillin V, then augmentin, and so on. Now, what are the species that is used, or which species is used? Now, the most common species that is used is Penicillium chrysogenum. Now, penicillin is a group of antibiotics which is extracted from the species of a mold Penicillium notatum. But for commercial production of penicillin, the species that is used is Penicillium chrysogenum. Here, this is an same image. Penicillin consists of thiazolidine ring fused with a beta-lactam ring and a side chain. Now, the side chain varies in different types of penicillin. Different types of penicillins are used in treatment of different types of bacterial infections. Now, here that you see is the structure of penicillin along with the molecular formula. Coming to production of penicillin, penicillin is produced by aerobic fermentation process, that is, it requires oxygen in the whole process. Penicillin is a secondary metabolite and is produced during the stationary phase when the growth of the mold is inhibited by stress. It is produced by fed batch submerged fermentation in a stirred tank fermenter. This helps to dissolve the oxygen and mix the content in a fermenter, that is for even distribution of the nutrient, and so on. Now, coming to the process of penicillin fermentation, it involves four major steps. The first one is strain development, secondly, inoculum production, the third step, inoculation, and the fourth one is extraction and purification. Now, we'll look at each of these steps. Coming to the first one, that is the strain development. So, over here, we have this is the wild strain. So, at the end, by through various steps, that is via plating, and now what is done is it is exposed to X-rays, the UV light, and a mutagenic agent. So at the end of it, we get a mutant strain. Now this mutant strain is then utilized for commercial production of penicillin. Now this strain is used in the next step, that is inoculum production. So from the previous step, a stock culture is produced, and from there that stock culture is subcultured, and the spores are then suspended in water or dilute solution of non-toxic wetting agent. The spores are then added to a flask of wheat bran and nutrient solution, and it is incubated for 5 to 7 days at 24 degrees Celsius, and it is allowed to sporulate. Then the spores are directly inoculated in the inoculum tank. So after sporulation, it is kept, or the temperature is maintained at 24 degrees Celsius, and then these spores are inoculated into inoculum tank. Now comes the inoculation process. It can be done in three ways. That is, it can be either done via dry spores, suspension of ungerminated spores, or pre-germinated spores, or mycelial pellets. Now, what is inoculation? Introduction of pure inoculum in the production tank or fermenters is called as inoculation. Now, if the spores are inoculated in dry spores, the spores are directly blown deep into the medium, or a wetting agent such as sodium lauryl sulfate is used. If it is done by a second method, that is suspension of ungerminated spores, then the spore suspension in sodium lauryl sulfate solution is fed to the fermenter using spray gun or pipettes. The fermentation medium is then agitated and aerated for better mixing. And the third, if it is done in the third manner, it is via pre-germinated spores or mycelial pellets. Now these pellets are fed to the fermenter, that is after 2 to 3 days of spore inoculation. Now coming to the medium employed for penicillin production should be suitable to achieve certain steps, or it should be suitable for the production. Now for that reason, we have three main criterias. That is, it should be suitable for abundant growth of mycelium, then maximum accumulation of antibiotics, and thirdly, easy and inexpensive extraction and purification of the antibiotics. Now, the medium that is used for production of antibiotics is Jackson's medium. The composition is given over here. Coming to the fourth step, that is extraction and purification. Penicillin is produced during the fermentation process. Penicillin produced during the fermentation process is extracted and purified, and this whole process consists of three steps, that is separation of mycelium, extraction of mycelium, and treatment of crude extract. Now first, the mycelium is separated from medium using rotatory vacuum filter. Then the extraction of penicillin is done by counter current extraction method. After separation, the pH of the liquid is maintained to 2 to 2.5, and this is carried out, that is separation is carried out by the phosphoric acid or hydrochloric acid. The liquid is extracted using organic solvent. Now the extracted penicillin is extracted into water from butyl acetate, and the pH is elevated to 7 to 7.5. Aqueous solution is then acidified and re-extracted with butyl acetate. The purified penicillin is in the form of sodium penicillin, and the sodium penicillin is then treated with charcoal. And finally, the sodium penicillin is separated in crystalline form by crystallization. To summarize the whole thing, penicillin is used for treatment of bacterial infection. Now, the strain that is used for commercial production, the wild strain, is Penicillium chrysogenum. The fermentation of penicillin involves a few steps, that is strain development, inoculum production, inoculation, extraction, and purification. These are the references. Thank you.